All right, so today we're going to do a deep dive into something that's pretty important for anyone working with cells, cell thawing.
Yeah.
And we're going to be basing this deep dive on a guide by Assay Genie.
Nope.
They're a company that provides a lot of different resources and tools for researchers.
Right.
And they have this really in-depth guide to cell thawing, which on the surface might sound kind of basic.
Yeah.
But it gets really fascinating.
Oh, absolutely.
When you start to think about what's actually going on at that cellular level.
Yeah.
So to help us unpack this, we have a cell culture expert here.
Hi there.
Welcome.
Thanks for having me.
So maybe you can just start by introducing yourself and
Sure.
what your expertise is.
Sure. So I've been working in cell culture techniques for over 10 years now. And yeah, it's been a wild ride. Cell culture is really at the heart of so much research. Everything from drug discovery to understanding disease mechanisms. It all starts with having healthy cells to work with. And proper cell thawing is absolutely critical for that.
Yeah, for getting those cells to behave how you want them to.
Exactly. You don't want to, spend weeks or months on an experiment only to find out that your cells weren't viable from the start because of improper thawing.
Right. Yeah, it all comes down to that healthy starting point.
It does.
So let's sort of back up and start at the beginning.
Sure.
When we talk about cell thawing, I mean, what exactly is happening there? I mean, what's going on?
Right. So, cell thawing is basically the process of reviving cells that have been cryopreserved.
Right.
Which means they've been stored at these ultra-low temperatures to stop all their biological activity.
So like hitting the pause button.
Exactly. It's like hitting pause on life at the cellular level.
So then we're hitting play on those pause cells.
Yeah.
And I've heard the term rapid thawing used a lot, and I'm wondering if you can kind of explain why speed is so important when it comes to thawing cells.
Sure. Yeah. So, one of the biggest dangers with thawing cells too slowly is the formation of ice crystals.
Oh, okay.
Inside the cells. And you can kind of imagine these ice crystals as like tiny little shards of glass.
Oh, wow.
That can actually puncture and damage those delicate cell structures, those membranes and the organelles inside.
So they're coming back online,
yeah,
but with internal damage.
Exactly.
Yeah.
Yeah. And so rapid thawing is really the key here because it minimizes the amount of time that those cells are exposed to those potentially damaging temperatures.
Okay.
So ideally, you want to thaw your cells at a rate of 37 degrees Celsius per minute.
Wow. That's fast.
Yeah, it's pretty quick.
That's way faster than anything I do in my kitchen.
Oh, yeah, for sure. It's
I'm thinking of frozen chicken here.
Yeah, no, it's completely different. But yeah, that speed really helps to prevent those damaging ice crystals from forming.
Okay, so speed is key.
It is.
The guide by Assay Genie kind of breaks this process down into 4 steps.
Yeah.
So we've got preparation, the actual thawing process, dilution and transfer, and then finally culture and recovery.
Yep.
So let's kind of take each of those in turn.
Sure.
And starting with preparation, what's crucial here?
So with preparation, it's all about getting your workspace and materials ready before you even touch that vial of frozen cells.
Okay.
And one of the most important things throughout this whole process is aseptic technique.
Okay.
Which is basically just a fancy way of saying we're keeping everything sterile to prevent any contamination.
So like a surgeon prepping for an operation.
Exactly. You want everything to be clean.
Right.
And in its place, you want to pre-warm your culture medium to 37 degrees Celsius.
Okay.
So we don't shock the cells with any sudden temperature changes. And of course, you want to have all your tools ready. And sterilize pipettes, centrifuge tubes, gloves, the works.
Okay, so it's all about minimizing that risk of contamination.
Absolutely.
All right, so we've got our sterile setup. We've got everything pre-warmed. What happens next in the actual thawing process?
So this is where the rapid thawing comes in. You take that frozen vial, which is usually stored in liquid nitrogen,
Okay.
and you quickly transfer it to a 37 degrees Celsius water bath.
Okay.
And the key here is to gently swirl the vial to ensure even thawing.
Okay.
But you also want to make sure that the cap of the vial stays above the water level.
To prevent contamination.
Exactly.
Okay, so it's all about controlled and even warming.
Precisely.
And then once that ice is melted, we move on to dilution and transfer.
That's right.
And I see the guide mentions getting rid of something called DMSO.
Yeah.
And that sounds kind of intense. What is that all about?
So DMSO is a cryoprotectant,
Okay.
which is basically a substance that helps protect the cells during freezing.
Okay.
So it's essential for the freezing process, but it can actually become toxic to the cells at room temperature.
Oh, okay.
So as soon as the cells are thawed,
Mm-hmm.
we want to dilute them with pre-warmed culture medium
Okay.
to minimize their exposure to that DMSO.
So we're diluting to protect. Okay. And then is it spin cycle time?
It is. Yeah, we use a centrifuge to spin the cells down and form a pellet at the bottom of the tube.
Right.
And then we carefully remove the liquid above the cells, which now contains that diluted DMSO.
Okay.
And we resuspend the cell pellet in fresh, pre-warmed culture medium.
So it's like giving them a clean, fresh start.
It is, yeah. It's like a nice little spa treatment after their icy journey.
Wow. It seems like a pretty delicate dance between speed temperature control and sterility.
It is, yeah. It's all about finding that balance.
So we're not done yet, though.
Nope.
We still have this culture and recovery
That's right.
stage. So what's going on there?
So at this point, we're basically giving those newly thawed cells, the TLC, they need to bounce back
Okay.
and start growing again. So we transfer them into a culture flask and place them in an incubator, which is a controlled environment with the ideal temperature, humidity, and nutrients for those cells to thrive.
So they're back in their happy place.
Exactly.
So to speak.
Yeah.
But the guy also stresses this idea of post-thaw care. Yeah. So it's not like you just set it and forget it.
No, not at all. You need to keep a close eye on those cells, especially in the first few days after thawing.
Okay.
You know, you want to be checking things like their attachment to the flask, their viability, and their morphology.
Right.
Which is basically just making sure that they look healthy.
So it's like a post-op checkup.
Exactly. Just making sure that everything's healing well.
All right. So far, we've covered a lot of ground.
Yeah.
We've got the importance of speed. We've got the importance of really meticulous sterility. And we've got careful monitoring post-thaw.
Yeah.
But things don't always go perfectly, right?
Oh, of course not.
So what about troubleshooting?
Yeah.
What are some of the things that can go wrong?
Yeah. So even with the best technique, you can still run into issues.
Okay.
And one of the most common problems is low cell viability after thawing.
Like they're not waking up properly from their frozen nap.
Exactly.
Yeah.
And there can be a few reasons for this. Sometimes it's related to what we've already talked about, like slow thawing rates.
Right.
Prolonged exposure to DMSO.
Okay.
But sometimes it can actually be due to issues during the initial freezing process.
Oh, wow.
So if the cells weren't frozen properly, they might be starting off on the back foot even before you get to the thawing stage.
So even with a perfect thawing technique, those cells might not be viable. So it sounds like each step has a ripple effect.
It does, yeah.
One misstep can impact the entire process.
That's right.
I think we need to take a pause here.
Okay.
And we're going to come back to this idea of troubleshooting in part 2 of our cell thawing deep dive.
Sounds good.
So don't go anywhere.
It really is. It's kind of like a chain reaction.
Yeah.
You know, each step builds on the last.
So back to troubleshooting, we were talking about low cell viability after thawing.
Right.
What other red flags should our listener watch for?
So besides those things that we already talked about, you know, the thawing speed and the DMSO exposure.
Yeah.
Sometimes you can see problems with cell viability even if the thawing was done perfectly.
Oh, okay.
And this is often a sign that something went wrong during the freezing process.
Okay, so even with a perfect thawing technique, those cells might be starting off on the back foot.
The back foot, yeah.
What kind of freezing issues
would cause this?
There are a few things that can happen. You know, one common mistake is not checking the cell viability before freezing.
Oh, okay.
So you're starting with a population of cells that's already not that healthy.
Right.
Another issue could be using the wrong concentration of cryoprotectant or not having a controlled rate of freezing.
So there are a lot of details in the freezing process.
There are. Yeah, it's a very precise process.
It sounds like there's a whole other deep dive that we could do on just the freezing part.
Oh, absolutely.
Yeah.
But let's say our listener has run into this low viability after thawing.
Right.
What steps can they take to try to troubleshoot this?
So the first thing I would always recommend is to double check their thawing technique.
Okay.
You know, go back to the basics, make sure that they're following the recommended rapid thawing rate, that they're diluting and removing the cryoprotectant promptly,
right,
and that the culture medium is fresh and correctly prepared.
So it's like retracing your steps
Exactly.
to see where you might have...
Yeah, like you missed a turn somewhere.
Missed a turn, yeah.
Yeah.
And if the thawing process seems solid,
right,
then it's time to go back and look at the freezing.
exactly.
Yeah.
Yeah, you want to review those cryopreservation methods.
Okay.
Make sure that everything was done according to protocol.
Now, besides low viability,
Yeah.
are there any other common thawing headaches that our listener might encounter?
Another big one is contamination.
Oh, right.
You know, we've talked a lot about how important aseptic technique is.
Mm-hmm.
But even a small slip-up can introduce bacteria, fungi, or other microorganisms into your cell culture.
And that's bad news.
That's very bad news.
Yeah. Like thinking you have the place to yourself and then suddenly realizing you have uninvited guests.
Exactly.
Yeah.
It's not the kind of party you want in your cell cultures.
So how can our listener prevent this contamination during thawing?
So it really comes down to being super careful.
Okay.
You know, always work in a designated clean cell culture area. Wear gloves and a lab coat to minimize contamination from yourself. Use sterile pipettes and tubes.
Right.
And be really vigilant when handling the cryovules.
So no swimming for the cryovule?
No swimming, no swimming. Yeah, keep that cap above water.
Okay.
And also make sure that your water bath itself is clean and regularly disinfected.
Okay, so we've got speed, we've got aseptic technique, we've got careful post-thaw monitoring. Anything else that our listeners should keep in mind for cell thawing success?
One more thing that I would emphasize is documentation.
Oh.
It's really important to keep detailed records of your thawing process.
Yeah.
You know, things like the cell line, passage number, the date of thawing, any observations you make about the cell viability and morphology.
So it's like a cell journal.
Exactly.
Yeah.
And this documentation can be really helpful for troubleshooting problems down the line.
Okay.
And it also ensures consistency in your experiments.
This has been a really thorough look at cell thawing.
It has.
It's amazing how much detail goes into something that seems
it's
so simple on the surface.
Yeah, it seems like it should be straightforward.
Right.
But there's actually a lot to it.
Before we wrap up, I do have one final question.
Okay.
The Assay Genie Guide mentions that cryopreservation is used in other areas, like biobanking and regenerative medicine. And it makes me think about how far this technology has come and where it could go next.
It really is remarkable. You know, the potential applications of cryopreservation go way beyond just cell culture. You know, we're just beginning to explore what's possible.
I mean, imagine being able to cryopreserve organs for transplantation.
Exactly. Or even entire ecosystems.
Wow.
To protect biodiversity.
That's mind-boggling.
It is. It's pretty amazing.
We're talking about potentially changing the world.
Yeah.
With these tiny frozen cells.
Exactly. And, you know, with that power comes a responsibility to use it wisely.
Right.
You know, as scientists, we need to think carefully about the ethical implications.
Yeah.
And the potential consequences of this technology.
It's a reminder that science is about more than just technical skills. It's also about our responsibility to use our knowledge for good.
I agree.
This deep dive into cell thawing has been incredibly insightful.
It has been. It's been a pleasure.
Thank you so much for sharing your expertise and your insights.
You're welcome.
And to our listener, we hope this deep dive has given you a comprehensive understanding of cell thawing.
Absolutely.
From the practical techniques to the broader implications of this field.
Yeah.
We've covered a lot of ground today.
We have.
So let's take a quick pause.
Okay.
Before we jump into the final part
That's good.
of our cell thawing deep dive.
You do.
All right. So we're back for the final part of our cell thawing deep dive. And, you know, we've talked about so much already, but it's really amazing to think about how much has to go right to bring these cells back to life after they've been frozen.
Yeah, it really is like this delicate dance of temperature control and sterility and timing.
And, you know, as we were discussing before the break, the applications of cryopreservation go way beyond just basic cell culture. I'm still kind of reeling from the idea of cryopreserving entire ecosystems.
I know, it's mind-blowing to think about.
It sounds like science fiction.
It really does, but it speaks to the truly remarkable potential of this technology.
Yeah.
You know, we've come a long way from simply preserving cells for research.
Yeah.
We're now looking at possibilities that could have a profound impact on healthcare conservation and even space exploration.
Yeah, I mean, it really makes you wonder what the future holds for cryopreservation.
That's the exciting part. We're constantly pushing the boundaries. Researchers are exploring new cryoprotectants, refining freezing and thawing techniques, and even investigating the potential of vitrification.
Vitrification.
It's an ultra-rapid freezing method that could further minimize cell damage.
Wow.
So, it's like we're on the cusp of a whole new era in cryopreservation. Who knows what amazing discoveries await
I
us?
know. It's exciting to think about.
It is, but as with any powerful technology, we have to proceed with caution and careful consideration. The ethical implications of cryopreservation are complex and constantly evolving.
That's a really good point. It's a good reminder that scientific advancements should always go hand in hand with those ethical considerations.
Absolutely. As scientists, we have a responsibility to not only push the boundaries of knowledge, but also to ensure that our work is conducted ethically and with the well-being of humanity and the planet in mind.
That's well said, and it's been an incredibly thought-provoking conversation. So thank you so much for sharing your expertise and your insights with us today.
It's been my pleasure.
And to our listener, we hope this deep dive has given you a comprehensive understanding of cell thawing, from the practical techniques to the broader implications of this fascinating field.
Remember, those tiny frozen cells hold the potential for incredible breakthroughs. And by mastering the art and science of cell thawing, you're playing a key role in unlocking that potential.
And on that note, that's a great point to end on. Thanks again for joining us on this deep dive into cell thawing. Until next time, keep exploring, keep learning, and keep those cells thriving.